Thank you for inviting me. Today, I'll be presenting my research that has already been published some couple of months back. So it's titled Profiling and Molecular Docking Analysis of Natural Bioactive Compounds Targeting Oncogenic Biomarkers in Breast Cancer, because especially we are talking about the breast cancer. So we have done some computational study with some phytochemicals to basically explore the phytochemical compounds that might serve as a safe and effective alternatives, or we can say that complementary agents in the breast cancer therapy. So starting with the introduction, as we are all aware that breast cancer is the most commonly diagnosed cancer in the women and a leading cause of cancer-related deaths if we talk about globally. So existing therapies are continuing. There is a growing demand for affordable, low toxicity therapeutics that can basically halt or reverse metastatic spread. As we are being continuously involved with the natural compounds to see their preclinical and their clinical effect. So they show the low toxicity, high safety, and can relatively target cancer cells, while sparing the normal tissues. So in this study, we have taken some of the phytochemicals such as berberine, curcumin, vitharin A, and ellagic acid, because they are cost-effective, abundant, and we say that sustainable. Okay, so coming to the study design and rationale. Actually, this slide summarizes one slide back. That is study design. Yeah. So this slide summarizes the core focus of our study, which is basically to investigate the inhibitory potential of 4 natural compounds, as I already discussed. It's curcumin, vitharin A, berberine, and ellagic acid, against key molecular targets, that are involved in the breast cancer. And these targets are basically BCL2, PD-L1, CDK4/6, and FGFR, and all of which play an important role in tumor growth, survival, immune invasion, and cell cycle proliferation. Progression. So to evaluate these compounds, we basically relied entirely on in silico approaches. We use the molecular docking to predict how strongly each compound basically binds to the target protein, and molecular dynamic simulation to analyze the stability of these interactions over time, and the tox analysis, that is ADME, to assess the pharmacokinetic characteristics. So overall aim, was to understand 4 major aspects, as how well these basically compounds they bind to the cancer-related protein, and whether if we proceed with these compounds, whether these interactions they remain stable under the physiological conditions, and their suitability as a drug candidate in terms of absorption and metabolism, and any toxicity concern that could basically limit their therapeutic use. So on this slide, basically we are outlining the core materials and methods here. So our primary goal, as I told you, it was about to explore the 4 phytochemicals and for their ability to inhibit the breast cancer biomarkers, and using the computational methods gives us a powerful advantage at the early stage of the drug discovery. So it is basically why we took the computational approach before taking it for the preclinical studies, because it's cost-effective, fast, and provides the high precision molecular information before moving to the laboratory experiments. And we obtained the 3-dimensional crystal structure of our target protein directly from the RCSB protein data bank, especially BCL2 from the structure that's already mentioned here to EUF, PDL1 from 3GQ1, CDK46 from 4MAN, and FGFR from 5J86. After downloading these structures, we basically prepared them using the PyMol, different softwares like PyMol, Chimera, and Swiss PDB. We were by removing the water molecules, correcting the structural and issues, and optimizing them for further analysis for docking and simulation. And at last, the phytochemical structure was basically retrieved from PubChem, energy minimized using FF94 force field and converted into another format, that's PDBQT, using OpenBabel for compatibility with software, docking software. So we also assess their drug likeness properties based on the Lipinski rule of 5 to ensure they meet the basic oral criteria for the oral drug candidates. For this, we basically used Swiss ADME for absorption and the bioavailability profile. This tool helps us to predict how well these compounds basically may be absorbed and distributed in the body. And additionally, it was also used to evaluate the toxicity parameters, including the hepatotoxicity, cardiotoxicity, and the mutagenicity. And then we performed the molecular docking, which allowed us to determine basically both the preferred binding configuration and the binding affinity of each compound towards the target protein. And to better understand this docking outcome, we visualized the ligand protein absorption using the BioAware Discovery Studio. And these tools basically help us to identify the specific residues involved and nature of their basically interactions. Besides, we also, after docking, we identified the hydrogen bonds and hydrophobic contacts and pi-pi skating with the key residues. For this, we did the post-docking and interaction analysis and selected complexes with the lowest binding free energy. We took it for the MD simulation. And to evaluate the dynamic stability of these complexes over the time, we performed actually 100 nanosecond MD simulation using this software, as we have already mentioned, the SMILE. And the simulations were run under physiological conditions using the TIP3B water model and OPLS 5E field force within NPT assemble at 1 atmospheric and 310 K. So we basically wanted to evaluate the complex stability and through RMST and RMSF profiles, which helped us basically to understand how the protein ligand complexes, they behave structurally, throughout the simulation. So coming to the results section, in this slide, I'm going to summarize the pharmacokinetic and the toxicity findings for our selected compounds. So first, the ADME pharmacokinetics evaluate how the compound is basically absorbed, distributed, metabolized, and excreted by the body. And these parameters, basically, we know that these parameters are basically very critical because even a high potent molecule, can fail as a drug if it has a poor absorption or a rapid clearance, I must say. So our ADME analysis helps predict whether these phytochemicals have a potential to succeed as a therapeutic agents, for the breast cancer therapy. And next, we also examined the toxicity profile to ensure the safety also. So this includes basically assessing the risks such as hepatotoxicity, cardiotoxicity, and the mutagenicity. So evaluating basically this toxicity early prevents advanced compounds that may cause other harmful side effects or adverse effects in humans, or they can pose other environmental risk. And so among the compounds that we have screened, as we took the 4 compounds. So particularly, attention was given to the 2 major compounds, that's berberine and the ellagic acid. So because both are basically known for their strong biological activity and the anti-cancer potential, as we can see, many research articles has been published on it, and the people have taken it, for different types of therapeutic options in both preclinical and then proceed to the clinical one. So our analysis showed that all basically these compounds, they demonstrated the favorable pharmacokinetic properties, and this indicates basically a lower likelihood, we can say, that in the later clinical studies, and highlights their potential as a promising candidates for further investigation. So besides it, we also did molecular docking of some of these bioactive compounds, and against the major targets that we took, the key proteins in the breast cancer, like BCL2, PD-L1, CDK4/6, FGFR. These basically proteins, they collectively regulate the apoptosis or immune invasion, cell cycling, and the signaling. So for this we use the molecular docking and visualization, to assess the binding affinity and the interactions of this. And the key findings that we had in the molecular docking for these bioactive compounds, where berberine basically showed the high affinity. Berberine have high affinity for BCL2, and with a docking score of minus 9.3 kilocalories per mole, so suggesting it may effectively inhibit the anti-apoptotic function of BCL2, and it also bound favorably to CDK4/6, indicating its potential basically to disrupt the cancer cell progression. And ellagic acid, it has higher affinity towards PD-L1, with a docking score of minus 9.8 kilocalories per mole, which may help basically block the immune invasion pathways. So it's also showing a strong interaction with FGFR, suggesting its interference with the growth factor signaling also. So across all the docking results that we did, we observed interactions such as hydrogen bond, pi-pi stacking, and van der Waals forces, which can basically contribute to the stable and biologically meaningful binding. So if I conclude this, so among all the compounds tested, we found that berberine and ellagic acid, they can be a promising inhibitors. They can show strong binding affinities, and they might be having a favorable interaction profiles, with the breast cancer biomarkers. And we can take them further for the preclinical evaluation. Lastly, we did a molecular dynamic simulation. Its purpose was to assess the stability and the conformational changes of the protein ligand complexes. And we performed to evaluate basically the stability and the behavior of protein ligand complexes over the time. And the focus here is on the 2 promising important compounds from our docking that we got from our docking result, that is berberine and the ellagic acid. And MD simulation allows us to basically observe how each ligand behaves within the binding pocket, under physiological conditions. And we evaluated basically the stability through several important parameters, as it's mentioned in the slide, like RMSE, to track overall structural deviations, RMSF, to assess the residue level flexibility, radius of gyration for compactness of the protein, and hydrogen bonds to monitor the key stabilizing interactions. And our findings were both ligands, basically, they maintain stable interactions with their respective target protein, throughout the simulation. Okay, then here also, and ellagic acid showed more consistent, RMST, minimal fluctuations, and higher complex stability. And berberine demonstrated the moderate fluctuation, as it's mentioned here, around 2 to 3 angstrom, but particularly between the 40 to 60 nanoseconds, and with higher conformational mobility. So both compounds form the stable complexes showing a slightly superior structural stability, insights, and the future, drug design. This slide basically summarizes the residue level flexibility analysis that was basically performed using the RMSF to understand how this protein, responds upon binding with two important ligands, that is berberine and the ellagic acid. And for this, we used the root mean square fluctuations to measure the mobility of individual amino acid residues, RMSF value, to identify which region of protein experienced the structural change during the simulation. And the findings were overall protein rigidity maintained and strong structural integrity during the ligand binding. And the highest fluctuation mainly basically occurred at N and C terminal regions, which is expected because these terminal ends are typically more flexible, and both protein and ligand systems demonstrated the minimal residue level movement, meaning that the atom migration remained low and binding interaction stayed the consistent throughout the simulation. So if we conclude, both ligands effectively preserve the protein stability, and there was a minor flexibility differences that may basically influence the biological activity. So here we did some secondary structure and the interaction analysis. We use this, and the key interacting residues were for the CID 5281855, we use some residues, and for the CID also we use some of the residues. And secondary structure content where with CID 5281855 protein maintained basically 34.27% helix, 8.01% strand, and totaling it was around 42.28. And for the CID 2553, the structural content was slightly higher. It was around 36.14, and strand with 8.37% strand, totaling around 44.51. So in conclusion, here also we found that both complexes, they remain stable. They maintain the stable secondary structure, and they show the strong persistent interactions with the key residues. And this stability further supports, their potential as a therapeutic candidate with, with neither ligand causing the significant structural differences. Then the binding energy calculation here, this slide summarizes basically the binding energy analysis, which was performed using the MMGB assay, which helps us to quantify basically how strongly each ligand, binds to the two UEF protein at an energetic level. And in the results, we found that berberine displayed a binding energy of around -78.14 kilocalories, indicating a strong and more stable interaction with the protein. And this large negative value basically suggests a higher favorable binding environment. And the ellagic acid shows a free binding energy of -57.32 kilocalories per mole, stable but less favorable than the berberine. And if we conclude it, so we found that the berberine basically showed the most energetic favorable interactions, highlighting its potential as a strong inhibitor of 2-UEF. And so if I finally, if I conclude it in a discussion, so this study basically provides a comprehensive computational evaluation of therapeutic potential of berberine and ellagic acid against key breast cancer biomarkers, including the apoptotic markers, and both compounds exhibited high binding affinity with berberine, showing particularly strong interactions, suggesting its potential as a potent inhibitor of oncogenic pathway involved in apoptosis, immune invasion, and cell proliferation. And pharmacokinetics and ADME profile indicate that both compounds, they possess excellent oral bioavailability, gastrointestinal absorption, and permeability, supporting their feasibility as orally administered therapeutic agents. So these favorable profiles reduce the likelihood of clinical failure due to the poor pharmacokinetics. Molecular dynamic simulation revealed stable protein ligand complexes with ellagic acid more displaying slightly higher structural stability, where berberine showed dynamic fluctuations that may contribute to diverse biological responses. If you take the RMSF value, RMSF analysis, it indicated that the ligand binding induced a minor local conformational changes without compromising overall protein integrity, and secondary structure analysis confirmed that both ligands, they preserve the helices, alpha and beta, highlighting the structural reliability under physiological conditions. Then binding energy calculation also collaborated with the two results, this methods that we already did, docking and MD simulation. They showed that berberine has a more energetically favorable binding profile compared to ellagic acid, so reinforcing its potential as a strong inhibitor of the breast cancer targets. So this slide highlights the future potential of our research. First, it points out the possibility of developing the dual target natural inhibitor, which could basically simultaneously act on the multiple disease pathways, enhancing the therapeutic effect. And next, integrating these compounds with the nanocarrier delivery system, it can improve their bioavailability, stability, and overall efficacy in the body. And these strategies basically open the door for precision oncology applications, allowing the treatments to be tailored to the molecular profile of individual patients. And this research approach can basically inspire novel combinatorial therapies, where natural compounds basically are combined with other drugs, to achieve the better synergistic effect and better clinical, outcomes. So for the future directions, we need basically to evaluate our findings, in vitro and in vivo, to ensure that the results are reliable and it can be reproducible. And it's important to investigate the pharmacodynamics and potential synergetic effects, which can help us to understand how these compounds basically, interact with the targets and with each other. And we should also explore, formulation strategies for enhanced delivery systems, such as the nanocarriers or other advanced system, basically to improve more bioavailability and the therapeutic, efficacy. So finally, to conclude the presentation, berberine and ellagic acid, basically, they emerge as a promising natural inhibitors for the breast cancer. They have a strong binding, stability, and low toxicity. That's basically confirmed computationally in silico studies. And it demonstrated the potential of in silico approach for the lead identification, and it encourages continued exploration of the phytochemical therapeutics. So I would like to acknowledge Al-Baha University and our collaborators for their support in making this research possible. It has been already published in scientific reports, and a special thanks to the Mathews International Conference on the Cancer Science and the Research for providing a platform, to share our findings. I'm also grateful to my research colleagues for the mentor for their guidance and contribution, they showed throughout this work. And last but not least, I would like to leave a message that together we move one step closer to the cancer-free world. Thank you for your attention.